I thought I'd start out by just telling you a little bit about myself and kind of my academic journey and how I ended up where I am today. And sometimes it's important for me to just remind myself too how I ended up here. Um, let's get this going here. Okay, so I'm originally from New Mexico. I was born and raised there. And I've realized more and more over the years that nobody makes as good a food as people do in New Mexico. So I go back there as often as I can, mostly just to eat. I grew up in a kind of a smaller town, but a small town that probably most people have heard of. It's Roswell. It's in the southeast kind of area of New Mexico, and it's famous for our UFOs and aliens, and strangely two TV shows about this little town that didn't seem all that exciting when I was a kid. But I go back there often because all of my family is there. So I grew up with a very large extended family in the same town as me. So I had, this is a picture of a lot of my aunts and uncles and cousins and my cousins' kids. And we all grew up together. In fact, next door to me lived my aunt and uncle and three of my cousins, and next door to them lived another aunt and uncle and three of my cousins. So we all just hung out together, and I had a lot of people that sort of helped raise me. And I had a lot of support, all along the way when I was a kid, and in a lot of different aspects. But nobody in my family, probably like some of you, nobody in my family had ever been to college or even thought about going to college. And so, when it came time to sort of figure out what I was going to do with my life, as supportive as they were, I don't think anybody really knew exactly, kind of what to do with somebody who wanted to go to college. And so I was a little bit on my own. And so you'll see a theme here coming out where, I just sort of made decisions along the way, a little bit, not really having much of a plan. So I asked some of my friends what they were doing, and a couple of them were like, "Oh, we're going to go to Colorado and go to school there." I was like, "Okay, that's cool." So I applied to one school. It was the Colorado State University, up in Fort Collins, and I got in, and I decided to go there. So I lived with one of my friends in the dorms there, one of my friends from Roswell. And I, thought I was going to go to med school. So I was a biological sciences major, pre-med. And then partway through, I thought, yeah, actually, I don't really want to go to med school, but I don't know what else I can do with this degree. So I started asking around, and I had a lot of great mentors there. In particular, sort of strangely, in the philosophy department. So I had taken a few philosophy classes, and I had a lot of great mentors there. And I was talking to one of them, and he said, "Well, what about being a professor? I think you'd be a good teacher." Like, okay, sure, I'll try that out. And so he helped me get into a summer research internship program, sort of like the one you guys are in. This was the McNair Scholars Program. And I got paired with a great mentor there, Dr. Hickey, who was doing exercise physiology, something I didn't really know anything about, but he really took me in and mentored me and showed me what research was all about. And then he was able to get funding for me through this NIH diversity program for the rest of the year. And so I did about a year and a half worth of research in his lab. And he helped me figure out, what careers I could look into, how to apply to grad school. And these programs were also really instrumental for that, really giving me resources to apply to grad school and, take GREs and really just have great mentorship along the way, which was really sort of instrumental for my education. So when it came time to apply to grad schools, I didn't really know anything about any of the grad schools other than what, I had been hearing from the people that were helping me out. So I applied to 2 grad schools, and I got into one of them, and I was waitlisted on another. And so I ended up going to the University of Wisconsin. I had never been anywhere, east of New Mexico or Colorado. And so I didn't know what I was getting into, but my fiance at the time, who I met at CSU, had family in Wisconsin. So again, I was like, oh, sure, that sounds fine. We'll have some support there, and family is really important to me. So I went to Wisconsin. I joined the program Cell and Molecular Biology, and I had a few options to join programs there, and I picked this one because it had the most options for faculty, but also had the fewest requirements for classes. And so, that's how I made my decision. And there I also was lucky to have really great mentors again. I was in the lab of Anna Huttenlocher, studying cell migration, kind of in the context of cancer. And I had these opportunities and funding and fellowship that really helped me along the way too. And it looks like we have some other McNair scholars in the audience too, so that's great. And so, I finished my PhD in 2005, and I didn't really know exactly what I was going to do for a postdoc, but I knew I wanted three things. I wanted to do neuroscience. I wasn't doing neuroscience at all. I just thought it was interesting. It seemed a little scary to change fields from grad school to postdoc, but people assured me that it was okay and actually a good thing. And so I wanted to do neuroscience. I wanted to use an in vivo model, like a mouse model or something, because I was doing all cell culture in my PhD. And the third thing is I wanted to go somewhere warm because Wisconsin is incredibly cold. You can see from the picture, it's beautiful for a couple of months, but it's also very cold for many more months. So I needed to thaw out. And so I was looking for those three things. And again, I had great mentors, and including my committee, my thesis committee. I had a couple of people that, I still keep in touch with. They still write me letters for everything. And they were really helpful. And they gave me a couple of names in San Diego that fit all of the things that I wanted to get out of it. And so in 2006, I moved my family to San Diego, and I started my postdoc at the Scripps Research Institute, studying developmental neuroscience. And so the bridge was kind of that I was studying cell migration, and the lab I was going to was studying neuronal migration in the developing brain with using a mouse model. So it had everything I wanted. Again, I was able to get some funding on a diversity supplement, which really gave me, some more resources along the way as well. So that's where I started studying what kind of some of the things that I study now in my own lab. So in 2013, I came back to Colorado, and that decision was, I had a few options for where to go, and talked it over with my family. We really wanted to come back to Colorado. We loved it. It was close to family, and had really great science, as you all are finding out this summer. And so we were super excited to come here. I started here on the Anschutz campus in 2013 as an assistant professor. Recently I became associate professor. So I'm in the Department of Pediatrics. And I'm also part of several graduate programs here, the neuroscience program, cell biology, stem cells and development program. And then I currently have a student in the molecular biology program, and also a student from the neuroscience program just joined my lab. So, this is kind of the center of our world here, are the graduate programs. They sort of kind of help keep everything together and help, cross disciplines and bridge everything. So I really like the way that the research is set up here. It's really graduate student centric, and the graduate programs are really sort of, again, the center of everything here. This also has given me an opportunity to kind of give back to some of the same programs that helped me along the way, right? I had really great mentors, as I mentioned, along the way that helped sort of shape my career trajectory. And that's probably my favorite part of this job, is kind of giving back in that regard and being a mentor. So I'm a part of a couple of the same programs that helped me out. So the SACNAS program, I go to the Abercams meeting every year. And recently I've been part of this really cool program that started out called Científico Latino, that pairs mentors with undergraduate students who are looking to apply to graduate school and kind of help them through the process the same way people help me through the process. So this has been a really fun part of my career as well so far. Great. So if anybody has any questions about any of that, I'm happy to answer it now. Otherwise, we can chat about it at the end too. I'll try to save a little time. Great. So for the science, I mentioned along the way that I became interested in neuroscience and mouse models and sort of bridged that to the cell migration world. And I started out in developmental neuroscience, and I'll talk about some of the things that my lab has been interested in. But mostly I'll focus on one story today on how we can, how neural stem cells know what kind of cells to make in the developing brain. All right, so, because I got my training in cell and molecular biology, I still tend to think of developmental biology and neuroscience as a series of kind of interconnected, overlapping cell biological processes. And so when I think of how the brain develops, I think about them in these different kind of steps. So, everything from the early kind of patterning of the central nervous system and how you set up different domains that become different parts of the central nervous system, to then what I'll talk about today, how the neural stem cell pools get specified into different cell types that go into making up this complex circuitry of the brain, how those stem cells balance proliferation versus differentiation. And I think you'll notice that a lot of these things are not unique to the brain, right? Anytime you're trying to build any tissue in or any organism, you've got to these are some of the questions that probably a lot of you are thinking about in your labs this summer, working on stem cells. How do they know whether they should divide or differentiate into another cell type? So we think about that a lot. And then once those cells are born, once those neurons and glia are born, they have to connect with each other. So send projections, they have to move to the right places, start to differentiate, and then form circuits. So these are kind of the things that we think about in my lab, how these happen during development. Half of my lab in the past has sort of focused on these phases down here, the later phases of development. This is kind of where I started with cell migration. And we've sort of extended some of those studies into once the cells get to where they're going, how do they differentiate and take on their final shapes and connect with each other. So these projects were really spearheaded in the lab by a CSD PhD student, Mark Gutierrez, who graduated last year and is now a captain in the US Army Medical Division. So that was kind of his career goal that he was really working towards, and he made that happen. So Mark was really working on the mechanisms of neuronal maturation. He had help from a master's student intern in our lab, who then became a PRA, and she's now at medical school, Lena, and our lab manager, Brett, who moved back home to Iowa last year. So we don't currently have anybody working on these stories, but we're still interested in them. Right now, what we're kind of focused on are these earlier stages in development, how the brain gets patterned and how those stem cells know what kind of cells to make. And these projects were started by my two very first students in the lab, Santiago and Caitlin, both CSD students who graduated a couple of years ago. Santiago was working on how neural stem cells know which kind of neurons to make. He's now a postdoc at Seattle Children's Research Institute. And then Caitlin was focused on some non-neuronal cells and how stem cells know not to make neurons, but to make glia, specifically oligodendrocytes. And I'll talk about her project a little later. Caitlin's actually still on campus and we collaborate a lot. She's part of the RNA Biosciences Initiative. She's a bioinformatics fellow. And I'll talk a little bit about that and some of her work at the end. And Luli is a second-year molecular biology PhD student who kind of has picked up where Caitlin has left off on this project. And all of these, again, kind of focus on what determines different fates in neural stem cells. Great. And so that's kind of the big question that my lab is interested in. To build functioning circuits, especially these complex circuits in the brain, they require a lot of different cell types, right? A lot of different kinds of neurons, but also non-neuronal cells that participate in the circuitry, like astrocytes and oligodendrocytes. And I'll kind of focus on oligodendrocytes today. And one of the big questions is that we're trying to address is, what are the developmental origins of all this diversity? In other words, how do this kind of uncommitted pool of neural stem cells or pool of uncommitted neural stem cells then know what kinds of neurons and glia to make and at what times in development and in how many numbers and in the right places, okay? So that's kind of what we're trying to get at. We use a mouse model, the mouse forebrain as our model, which you can see here highlighted in this green transgenic embryo here. So if we take a little cross section through the brain here, it'll look something like this. And one of I'll talk about two kind of main themes in the field for how you generate diversity in the forebrain. One is regionalization. So different parts of the forebrain get set up to make different kinds of cells. So for example, this blue part up here, the dorsal forebrain. So again, this is a cross section. So dorsal would be up here. This blue domain are progenitor cells or stem cells that are going to make excitatory neurons. These are neurons that are exciting each other when they connect to each other. And they make all of the excitatory neurons for the cerebral cortex. Conversely, this green domain down here, this ventral part, gets patterned really early on in development. And this green pool of stem cells gets set aside to make a different kind of cell. These are inhibitory interneurons that move up into the cerebral cortex, and they provide the inhibition in the circuitry. All right, so regionalization is really important. But even within these pools of regionalized progenitors, they make lots of different cell types. And so another way that you can generate diversity within a pool of progenitors is by timing. So we know that if you look at just the different excitatory neuron subtypes, there are neurons down here in blue that project down to the thalamus. There are neurons here in red that project down to the spinal cord. These ones in blue and green project across to the other hemisphere. And we know that these different cell types are born at different times in development. So, for example, these ones that project to the thalamus are born first, and then later on, the ones that project to the spinal cord are born. And so within the same regionalized pool, you can generate diversity through timing as well. Okay. And in the end, I'll kind of bring these two things back together. All right, so if we look at the cell types that are made in the developing forebrain, which again is our model system, we have these stem cells down here. They divide to make more of themselves. At some point, they kind of transition into this progenitor phase where they can start making neurons, these early born neurons, and they can self-renew. They make an intermediate precursor type that will generate other kinds of neurons, and they can self-renew. And then later in development, this progenitor pool starts making glial cells, astrocytes, and oligodendrocytes. So over time, they're making different kinds of cells. And these are all the cell types that the progenitors it's actually not all the cell types, it's most of the cell types that the progenitors in the developing dorsal forebrain can make. They mostly make excitatory neurons, and they go through this intermediate progenitor cell phase. A lot of them, after they make neurons, transform into astrocytes. These are glial cells that participate in a lot of the functions of neurons. They're really close to synapses, and they help regulate the connections between neurons and the health of neurons. And then towards the end of development, they can also make, a subset of them can make oligodendrocytes, which I'll talk about in a minute. This is another glial cell type, and they go through an oligodendrocyte precursor phase. And they can also make a different kind of neuron that migrates to a different place in the brain, called olfactory bulb interneurons. Great, so I'm going to talk about oligodendrocytes. So many of you probably have heard of oligodendrocytes. They're a glial cell type they reach out. So here's the cell body here. They reach out onto all of these neuronal axons, and they start wrapping them with sheets of fatty myelin that kind of acts as an insulation for these axons and helps propagate action potentials down the axon. So oligodendrocytes are really important in the circuitry because they help with the timing of when neurons fire to each other. They can speed up or slow down the speed of with which the signal goes to the next neuron. So they're really important for proper communication in the central nervous system. These are the cell types that are affected in multiple sclerosis. And so these axons don't really like to be naked or uninsulated. And so when the immune system attacks the oligodendrocytes in disease, and this myelin goes away, the axons are not happy, the circuit is not happy, and eventually these neurons can degenerate and die. And so, one thing that I think probably a lot of you are interested in is, potential stem cell therapies. I think in the brain, the oligodendrocytes are a really interesting target for potential stem cell therapies or, stem cell transplantation to generate more of them because they are lost in certain diseases, and they're pretty good at, when you put them back, at knowing what to do and where to go and which cells to connect with and myelinate afterwards. So we're trying to learn about how oligodendrocytes develop and function in a normally developing mouse brain, but part of the idea is that eventually, if we learn enough about them, we might be able to understand how they are dysregulated in human disease and be able to treat or prevent that. Okay, so we know a fair bit about how oligodendrocytes develop from this precursor stage down here, how they mature into and to make myelin. We don't know a whole lot about where they come from initially or how they're generated. And so that's kind of what I'll talk about today. How are these oligodendrocyte precursor cells made from these progenitors in the cerebral cortex? And how do these progenitors know to make OPCs and not all these other cell types? I don't know what's going on with this slide, so I'm going to skip it. So when Caitlin first started in my lab as a PhD student, she was really interested in this problem, and I hadn't studied oligodendrocytes at all. This was a question she was interested in. We were studying how these progenitors make neurons, but Caitlin realized there was this gap in the field where nobody was really looking at how progenitors decide to make oligodendrocytes. And Caitlin knew that in other contexts, like the spinal cord, this pathway here, called the sonic hedgehog pathway, is really important. So this pathway is named for this secreted protein here called sonic hedgehog. It's a ligand for this receptor patched. And normally this receptor patched inhibits this other protein called smoothened. But when sonic hedgehog binds to patched, smoothened gets activated, which then turns on these transcription factors called GLI, which go in and drive transcription. And the sonic hedgehog pathway is sort of one of these classic pathways that seems to do everything. It's involved in developing limbs and developing patterning all over the embryo. And it was known that somehow sonic hedgehog signaling was involved in generating oligodendrocytes in the spinal cord. So Caitlin thought maybe this pathway is also important for generating oligodendrocytes in the developing forebrain. Maybe it's helping tell progenitors how to make oligodendrocytes instead of other cell types. So she tested this using a genetic model, and I won't get into the details here, but this is just to illustrate that we can knock out this smoothened gene, which then disrupts this entire pathway, specifically in the progenitors that we cared about up here in the dorsal forebrain. All right, so all of these green progenitors up here will be smoothened knockouts, and we'll just simply ask, can they still make oligodendrocytes or not? And the answer was no, not really. So normally at embryonic day 17, these progenitor cells start to make lots of oligodendrocytes. We can label the oligodendrocytes in a brain section using this, an antibody against this protein called OLIG2, which is essential for making oligodendrocytes. So we see all of these oligodendrocytes. We're just taking a little snapshot of one piece of the developing brain right here. But in this piece, you can see all of these oligodendrocytes. When Caitlin knocked out smoothen to block sonic hedgehog signaling, we had a lot fewer oligodendrocytes present. And actually the ones that stick around are from a different part of the brain that moved in, and they happen to still have smoothened in them. So most of the progenitors where we knocked out sonic hedgehog lost their ability to make oligodendrocytes. We also collaborated with Sam Pleasure's lab at the UC, I don't know where they are, I forget, sorry, it's early. Anyway, we collaborated with a lab that did the opposite experiment. Normally sonic hedgehog signaling is low earlier in the brain and you get no oligodendrocytes. But if we knock out this negative regulator called SUFU, sonic hedgehog signaling goes crazy. And now we get a lot of oligodendrocytes down here when we don't normally have oligodendrocytes. So not only is sonic hedgehog signaling necessary for making oligodendrocytes, it's also sufficient to make some at a time when they wouldn't normally be making them. All right. So we're kind of building this model where here towards the end of development, when these progenitor cells are about done making neurons and they should switch to making oligodendrocytes, we think sonic hedgehog is important for this. We also showed that if you bring sonic hedgehog to them a little too early, they will make oligodendrocytes at a time when they should be making neurons, right? So sonic hedgehog somehow triggers oligodendrogenesis. This was kind of weird to us because the sort of dogma in the field was that there was no sonic hedgehog in this part of the brain at this time. That if, you know, we knew that sonic hedgehog was important in the ventral part of the brain to set up that patterning that I told you about, that sort of regionalization where you have a dorsal part and a ventral part. Sonic hedgehog was important for making the ventral part. But we're talking about the dorsal part here, and there shouldn't be much sonic hedgehog if that was, you know, how things were working. So Caitlin wanted to know, where's the signal coming from? And I had a great idea that I thought was really cool, which was that, you know, these progenitors up here in the dorsal forebrain that are labeled in blue up here, they must be secreting just a little bit of sonic hedgehog. And this could serve as a timing mechanism where as you produce more and more neurons, that little bit turns into a lot. And then, you know, that tells them, okay, we have enough neurons, you can start making oligodendrocytes right now. So Caitlin tested this and it turned out to be completely wrong. She knocked out the sonic hedgehog ligand from these cells up here, and everything was fine. The progenitors made oligodendrocytes with the right timing and in the right numbers. So Caitlin had a better idea. She thought, you know, there's another cell type that comes into the brain just before oligodendrocytes do. And they're called interneurons. You can see them here in green. They're actually born down in these ventral regions, which have high sonic hedgehog, and they migrate really long distances up into the dorsal forebrain, which has low sonic hedgehog. And they migrate right next to these progenitors up here. And so Caitlin knocked out sonic hedgehog from these interneurons. And sure enough, she saw that then these progenitors up here were no longer able to make the right numbers of oligodendrocytes. She also found a secondary source of sonic hedgehog. There's this little structure down here called the choroid plexus, which secretes cerebral spinal fluid into this space here. And they can also secrete sonic hedgehog. And so these two combinations of sources were what was bringing the sonic hedgehog up to the progenitors at a time just before they started making oligodendrocytes. I'm going to skip over this. It's just a sort of summary.
Santos, you have a question in the chat from
Cool.
Jimmy. He's curious to know if mouse models also show similar trends in disease states, such as beta amyloid plaques and diseases, Alzheimer's, dementia for humans. And then he goes on, he remembers sonic hedgehog pathway signaling from Dr. Barlow's seminar shows a couple of weeks ago. It's interesting how sonic hedgehog is intertwined with dorsal neural signaling separately, as well as the sense of taste loss that we went over with the seminar with Dr. Barlow.
Yeah, that's great. Those are great questions. And yeah, I'll sort of address the second one first. It is kind of interesting how like we pick up all of these different ways that the same molecule or the same pathway can regulate cell fate in different contexts. I mean, you wouldn't think that making a glial cell type in the brain would use the same mechanisms as making, you know, taste buds on the tongue, but it is. And I think it's always fascinating to see how the same pathways get used for different processes. And, you know, I think there's something to be learned from that too. You know, what are the common mechanisms, and can we think about cell fate in the brain in a similar way as cell fate, in taste buds. And that's what's really cool on this campus is we have a lot of people sort of, speaking similar languages in terms of their science, but in different contexts. And that's really fun. Joe's lab and my lab study very similar things, but in different contexts. And, we have meetings together, and we have journal clubs together. And so that's been really, really fun. And one of the reasons I actually decided to come to this campus is sort of this really collaborative nature that we have here. Yeah.
Can I add one thing there, Santos? Yeah. So Santos mentioned a model of how Sonic Hedgehog could be affecting oligo development by little bits secreting to feed back. And actually in the retina, Sonic Hedgehog does that model. And so it's pretty neat how in one context it does one thing and in another it does something slightly different. Same basic signaling pathway. It's very cool.
Yeah, totally. And then Jimmy, your question about mouse models, I think that's one great thing about, in particular, nervous system development is that it is really well conserved between mice and humans. Mice are not just, they're not tiny humans, so there are plenty of differences, and those differences are really interesting too. So it's kind of cool to study the differences as well. But yeah, so disease states, we can get mice to have demyelinating lesions that we can then see them repair, affect oligodendrocytes and get neurons to degenerate and then bring them back. So it's a great model for testing some of these therapies or preventative measures too. There are people on our campus that do these kinds of things. But, you mentioned beta amyloid plaques in diseases like Alzheimer's and dementia. Some of the cellular mechanisms and developmental mechanisms we can really see, and some of the aging mechanisms we can too in mice. Some other things are a little bit harder, like, it's a little bit harder to tell if a mouse might have dementia. Some of the behavioral aspects I think are a lot trickier, but people study those too. So I think they are very good models, but I think, there are plenty of ways that they might fall short as well. And I think it's important to kind of recognize those shortcomings as well and try to figure out other ways to get at that. You'll probably hear talks here in this seminar series, or maybe you already have, of people using, human cells to make organoids, things that sort of recapitulate development, but using human cells. And that might be another way that we can get at some similar kinds of things, but not using rodents. Cool. Great. So, we started to sort of add to our model where not only is Sonic Hedgehog important from at this time, but we know what kind of cells are bringing them in. They're coming from these interneurons and also from this choroid plexus. And so this is the model that Caitlin came up with, which we're still working on. We know that early in development, these progenitors down here in yellow are making neurons. So they're undergoing neurogenesis. And later in development, some of these progenitors start making these oligodendrocyte precursor cells, which go on to make oligodendrocytes. So by the end of the mouse embryonic development, or around when they're born, they're no longer making neurons. They're making almost all oligodendrocytes and astrocytes, so glial cells. We're really interested in this transition period because this is a period where some progenitors are still making neurons and others are making oligodendrocytes. And we're interested in understanding what's different between these progenitors to try to get at the instructions that are important for telling them to make either neurons or oligodendrocytes. We don't yet know if it's the same progenitors that can make neurons and then oligodendrocytes, or if these are totally separate stem cell pools that are just kind of mixed together. That's a question we're really interested in. But over the last several years since Caitlin first published this story, we've really been focused on this. How do these yellow progenitors know to make neurons and these green ones know to make oligodendrocytes? And that's something that Luli has continued on as well, and Caitlin has continued working on this. So I said I'd bring sort of this timing and patterning back together at the end. We think this is what's going on with Sonic Hedgehog, that early in development, when these progenitors up here are making neurons, Sonic Hedgehog is really important for regionalizing those stem cell pools. and it tells these ventral pools to make a different kind of neuron than these dorsal pools. But then later in development, these ventral sources sort of infiltrate the dorsal forebrain and they bring sonic hedgehog with them. And this is important for the switch from making neurons to oligodendrocytes. And so, we think that regionalization and timing can actually cooperate here to promote this neuron to oligodendrocyte switch. Great. So again, this question that we're really interested in is how does sonic hedgehog drive some of these progenitors to an OPC fate? So we're sort of digging in now and asking, okay, we know sonic hedgehog is important, but how? What is it doing to those cells? And so one way we've started to get into this is through Caitlin's training in bioinformatics. This has been, really popular in the field lately, and even on our campus, a lot of people are working on using single cell RNA sequencing to get at some of these questions. And if you're not familiar with this, is the way to take individual single cells like these progenitors here. So, oops. Individual progenitor cells here, and then use sequencing technologies to figure out what genes are being expressed in those cells in a single cell, and at what levels. And so you can ask between this cell and this cell, what's different between them, or this pool of cells and this pool of cells, what's different from them, which is perfect for the question that we want to ask is what's different between those progenitors making neurons and the ones making oligodendrocytes. And then we can use those differences to try to get at, what the instructions are that tell them to make those different kinds of cell types. So this is just, I'm not going to go through this data. This is just a representation of that we can, we've actually been analyzing other people's published single cell RNA-seq data sets. Other people on our campus are doing this themselves. So far, as part of Caitlin's training, she's been taking data sets from other labs that are published and reanalyzing them and asking different questions than what they were asking. And so she gets these cool little maps where the individual cells are in different clusters, and those cells in each cluster represent cells that are more similar to each other. They're expressing the same genes. And she can pick out the progenitor cells and the oligodendrocytes and just subset them. And she can ask, what kinds of genes are differentially expressed in the oligodendrocyte lineage compared to the other, let's say, astrocytes or neuron lineages. And so these would be individual genes up here. And we've been particularly interested in this one transcription factor called ASCL1, which comes on in progenitors that look like we think they're going on to make oligodendrocytes. And part of that is through Caitlin's analysis, where you can do these really cool analyses where you can ask, based on the similarities or differences in what genes these cells express, can we predict that a pool of progenitors is going on to make one cell type instead of another based on their gene expression? So again, this is all bioinformatics. These aren't, none of this is like actual lineage tracing experiments yet, but that's what we would follow up with. And she can predict lineages that go through different clusters that go on to make oligodendrocytes versus neurons. And she can project them over like a pseudo developmental time based on their gene expression. And again, we really focused on this transcription factor ASCL1 because if you analyze the sort of predicted activity of this transcriptional network, it looks like it's really important for making oligodendrocytes, and this gene stays on a lot longer in oligodendrocytes compared to neurons or astrocytes or other kinds of cells. And so that's what we've been kind of focusing on lately. Luli in the lab has followed up with that and showed that ASCL1, we knew already that it was really highly expressed in the ventral forebrain down here. But if you zoom in on this little box, she showed that ASCL1 RNA is actually expressed in these progenitors down here, which would be up in the dorsal forebrain. And as these progenitors start making oligodendrocytes, ASCL1 levels increase up here. Caitlin had actually shown that just like knocking out smooth-end gets rid of oligodendrocytes, it also decreases the ability of these progenitors to make ASCL1. And so sonic hedgehog seems to be important for ASCL1 expression as well. And I'm not going to go into this data, but this is just to say that we use some genetic tricks to label the ASCO1 positive progenitors and then later ask what kinds of cells they make. And a large proportion of them do make oligodendrocytes. All right. And so, using bioinformatics, we've sort of followed up on Caitlin's initial studies and showed that ASCO1s might be functioning downstream of sonic hedgehog. It's expressed in the right place at the right time. If you knock out sonic hedgehog, ASCO1 expression goes away. And a lot of these ASCO1 positive progenitors make oligodendrocytes. And so now we're trying to ask, how is it that ASCO1 is driving the decision to make oligodendrocytes and not neurons? And that's something that Eliza in the Gates Summer Internship Program is working on in the lab. We're using some genetic tricks and some other approaches that we have in the lab to really dissect the role of ASCO1 in this process. So the model that we're working with right now is that ASCO1 works against and with different transcription factors in these different lineages. So other people have shown, other people in our other labs in our field have shown that this transcription factor neurogenin-2 and this transcription factor TBR2 are important for telling these progenitors to make these IPCs and eventually excitatory neurons. And a lot of studies have shown that neurogenin-2 actually inhibits the expression of ASCO1. The model that we're kind of working with, and it's even changed since I made this slide, is that sonic hedgehog somehow comes in and is trying to drive this oligodendrocyte fate through this transcription factor OLIG2. And the job of ASCO1 in this might be to inhibit this alternate fate, to inhibit these other transcription factors. And I just don't have time to go into any of this data, but like I said, Eliza is really sort of working on this project now, trying to understand, how do sonic hedgehog and ASCO1 really work together? Are they in this linear pathway? Are they cooperating? What's going on there? And this is part of a follow-up manuscript that we're writing up and we're going to submit this summer. Great, and so that's all I have for you today. I just want to again kind of acknowledge the people that are doing this work. Caitlin started this project working on sonic hedgehog and oligodendrocytes at a time when we didn't work on either of those things, and I actually knew nothing about either of those things when I started my lab. So that was all driven by Caitlin and her interests. Luli has picked up this project. I don't have a picture of Eliza yet, but you see a picture of her there in your participants window. And she's working on this summer. And I don't have a picture of Maddie yet. She just joined the lab, well, actually officially today. And so I'm gonna have to update our pictures. Again, thanks to all of the departments and funding. I have a really great department in pediatrics, and our section of developmental biology is very supportive. A lot of great faculty there. The Children's Hospital Colorado Research Institute, of course, the Gates Center for Regenerative Medicine, some of our funding sources. And again, thanks for waking up early and coming to my talk today. Happy to take questions.